Hi everyone, welcome to this M2D2 talk. Today we are honored to host Pat Walter and James Thompson for the talk titled "There's no free lunch, but you can get a discount: Applying active learning in drug discovery." A bit about the speaker before we start. Pat is currently the chief data officer at Relay Therapeutics. His group specializes in developing innovative computational methods that merge computer simulation and experimental data to produce insights that drive drug discovery programs. Prior to Relay, Pat spent more than 2 decades at Bandit Therapeutical as the global head of modeling and informatics. Thank you, Pat, so much for joining us today. And James is a software engineer at Google. He is currently working on medical image segmentation as part of the applied science team. And prior to that, he received his PhD in computer science at the Rensselaer Polytechnic Institute, where he studied community detection approach in social network. Again, thank you so much, James, for accepting the invitation today. And both speakers are, because we have two speakers today, if you have any question, please write the question in the chat. And you taking question at the end because we have two speakers today. And yeah. So without further ado, just let jump right in. James?
Thanks. Yeah, well, definitely excited to be here and to be able to talk about some of this work. So as mentioned, we're going to talk a little bit about a couple of ways that active learning can be leveraged across different parts of the drug discovery process. And so the drug discovery process itself has many different phases you might be familiar with. At the beginning, you have kind of target identification, where you try to identify a protein whose behavior you want to modulate or change in some way. Then you have hit finding, where you try to find molecules that bind to the target at least a little bit. Continue on to lead optimization, where you try to refine those hits, and then so on and so forth, all the way down until hopefully you have a pretty good drug. In general, there are computational tools and machine learning models that have been used at various points, in all the stages to kind of help improve the efficiency and accuracy of drug discovery. But active learning is a more general technique, so it's something that can be applied to kind of all of the approaches, or any approach that you want to take, and it tries to help the methods use whatever data is available to them more efficiently. So specifically today, I'm going to do a little bit about active learning for FEP optimization, which is a little bit later in the process. It's lead optimization. And then Pat's going to talk a little bit about active learning for virtual screening. Cool. So for lead optimization, before we get to that, for lead optimization, as mentioned, we kind of build on work that was done earlier. So at this point, we have kind of a target protein that was identified during target evaluation, and we have some molecules that we know or assume bind to that protein, at least a little bit, that we found in the hit identification stage. The lead optimization stage, now we're trying to refine these hits and find kind of the best binder that we can using the information that we gained from the hit identification stage. So the straightforward way to do this obviously would be to kind of make and test a bunch of different molecules and see how they bind to the target protein. But as you can imagine, that's kind of a pretty manually intense and expensive and time-consuming thing to do. So active learning tries to kind of help mitigate this by reducing the number of molecules that you have to synthesize and test, while still being able to find or recover really good binders. So to kind of show an example of active learning before we get into molecules specifically, consider trying to find the best tasting wine, or your favorite wine, or a wine that pairs well with a specific meal. There are way too many wines that exist right now. It would be impossible to taste and try each and every one. So you kind of need a strategy to pick which wines you're going to try to taste and which ones you're just going to skip or ignore. Every wine is different, so every wine has different characteristics. Some are going to be more acidic than others, some are going to be sweeter than others, and so on. But they can be grouped together in kind of classes of wines. So you have Rieslings, Pinot Grigios, so on, and wines kind of within a group have similar tastes, similar characteristics, and so on. So one way to kind of track the search or track the search for the best wine is to kind of make a map of all of the wines based on the characteristics that they have. So if we only consider the two characteristics, say sweetness and acidity or whatever, the map might look something like this, where say sweetness is on the x-axis, acidity is on the y-axis. And if this is done correctly, the points of two wines that have similar tastes should end up kind of closer to each other than two wines that taste very differently. So as a consequence, the wines that are in the same category are going to bunch up together as well. So, reds might be Chardonnays, and blues might be Pinot Grigios, so on and so forth. Okay, so to start the search for the greatest wine, there's no information available off the bat as to which wines are going to be good and which ones are going to be bad. Let's say we know nothing, about wines. We can perform an initial exploration of different wines, just kind of trying to sample different things. So maybe we try, one example of each wine. Maybe we try some wines that are more acidic, some that are sweeter, so on and so forth. And the idea is that this would give us a kind of general idea of which characteristics might be good, which might be not so good, and sort of form a base that we can start from. Once we do this, we can kind of identify some wines that we like more than others, and then we can see kind of where those wines fall on the similarity map. So for argument's sake, let's just say, a Merlot and a Cabernet happen to be two of the ones that were that we enjoyed the best or that were the best tasting for the initial exploration. At this point, we can kind of switch gears a little bit and focus just on the types of wines that we found were good. So instead of just kind of doing a general sampling, we're focusing in on just Merlots and just Cabernets at this point. And if we did the mapping correctly, that kind of corresponds to exploring the points around the wines that we know are good. And hopefully, once we do this, we're kind of in the space of good wines, and we can kind of whittle down and start finding the best of the best. One of the drawbacks when we do this, though, is that we run the risk of kind of skipping or ignoring or missing good wines from other groups or clusters. So, for example, there might have been a Riesling or something that would be much better than Merlots or the Cabernets that we're trying here, but we're ignoring it because the initial Riesling that we had sampled was really bad. And that could have just been coincidence. We just might have had a bad example. On the other side of things, if we try to explore too much and keep tasting all these random wines, it might just become a waste of time. So there's the kind of practical limit to the number of wines that you can taste in any finite period of time. But there's also the point where if you've tried, 100 Rieslings and you don't like any of them, it starts to become probable that you just don't like Rieslings. So active learning specifically tries to take this sort of exploration, exploitation trade-offs and try to balance the strategies of exploring a space and exploiting the knowledge of the space to try to kind of find an optimal target, as efficiently as possible. All right, so back to the molecules specifically. Instead of finding, a favorite wine, here what we want to do is find a molecule that binds very tightly to a protein. So similar to the wine example, every molecule has characteristics, and we can use those characteristics to kind of map a molecule to a search space. We specifically use, molecular fingerprints, Morgan fingerprints, but we also experimented with RT-kit properties and any kind of molecular characterization you want to use here, as long as it's reasonable, should work. This specific picture is a mapping of 10,000 analogs of a known TYK2 binder that we took the fingerprints of, embedded in a space, and then projected into 2D. So it's a little bit condensed, but it gives a general idea of, you can kind of see some clusters of similar molecules and different spaces. So now the goal here is to find, the 1 or the 5 or however many molecules you want that are the best binders to TYK2. Again, synthesizing and kind of testing all of these molecules would take way too long. So we're going to try to use active learning to help us out so that we don't have to make all of them. In this case, kind of something that's different from the wine example is that we can use computational proxies to help us out. That might give us kind of an idea of what the binding affinity would be if we did experimentally test these. And kind of one example is free energy perturbation calculations, or FEP calculations. So there are a few different FEP calculations, but in the implementation we use, we kind of start with a known molecule as a starting point, and progressively kind of change or morph that molecule into the molecule that we would want to know the binding affinity for, so something we haven't seen yet. And then it uses molecular dynamics to kind of calculate the free energy of the transformation from the known molecule to the new molecule. In general, lower free energies signal kind of a higher binding affinity, so we're looking for lower numbers here, but this just kind of gives an estimate of what we would find experimentally. So with this, the task changes a little bit from finding the molecule with the best binding affinity to finding the molecule with the lowest FEP calculation value. So unfortunately, this does help with kind of efficiency of things, but it still takes a very long time to do. With the implementation we used, calculating a single FEP value for a single molecule with a single GPU takes about 4 to 8 hours. So if we were to try to calculate FEP values for all 10,000 in the set that we're using, it would take about 9 GPU years. So the calculation is embarrassingly parallelizable. So we were able to do it for this one data set, but in general, it's not something you'd want to do repeatedly if you had different target proteins, or you wanted to try different search spaces, or even search spaces that are a magnitude larger than what we have here. So millions of molecules probably wouldn't work. All right, so the last thing we need to kind of leverage active learning here is a way to choose which molecules to run FEP on and which molecules to ignore. So for this, we kind of add another layer of indirection or estimation and introduce a machine learning model which estimates the FEP calculation. So you have a machine learning model estimates FEP, which estimates binding affinity. In exchange for this extra layer of estimation, what we get is much quicker compute time. So instead of the years of GPU time that you would need for FEP, we can train and infer a very simple machine learning model on the 10,000 examples in CPU minutes or CPU seconds even. So it's much more practical. On the right, you can see a comparison of a model's predicted FEP with the actually calculated FEP. And there definitely is some error there. So ideally, you'd want the FEP calculation, but the model accuracy seems useful, at least. It has the right trend. Okay, so those are all the pieces. And this is kind of what the active learning cycle or process looks like as a whole. We start with our 10,000 molecules as a selection pool. We pick a set of them to run FEP on, get the actual values for those. We can then train a model on those values and use that model to infer FEP values for the rest of the molecules in the selection pool. With this new knowledge, we can then select a new batch, and hopefully the information that we got from the FEP trained model will help guide us towards better molecules quicker. It is possible to kind of train a model to do the initial selection as well if you have some other data set or some previous data set that you want to use. But for this data, or for this experiment, we assume we don't have that, and we just start kind of from ground zero. So we use other approaches like randomly sampling or things like that. Cool. So I guess when running this prospectively, one of the downsides of active learning is even if you find, say, like a molecule that you think is a really good binder, it's difficult to know how well the active learning did because there are some molecules you never saw, so you don't know if they're better or not. But we have the ground truth kind of calculated FEP values, so we can do in-depth analysis, and we can do things like track the model accuracy through all of these active learning cycles, or make sure that we did actually get the best molecule at the end of the day. Yeah, so cool. So diving into an example of one of these experiments, in this specific kind of iteration, we took a random sample of 50 molecules as our initial kind of batch or guess, and the red points on the left indicate where those molecules kind of fell in the selection space. So you can see even though it was just uniformly random, it kind of gives a general coverage of the space, which is really good. And then on the right, there's also a plot of the FEP values. So the average FEP value of this bunch happens to be just above 0, and it has a pretty Gaussian distribution. As a last note, kind of the measure of success that we used for this experiment was the number of top 100 molecules found. So those would be the 100 molecules in the set of 10,000 that have the lowest FEP scores. And it just so happened that in this experiment with this batch of 50, we sampled one of the top 100, so we got that one, more or less for free, which is a pretty nice start. Okay, so after the first round of active learning, where we trained a Gaussian process model on the initial 50 points, used that model to infer FEP values for the rest, and then greedily took the molecules with the lowest FEP value. We had 50 new points, and so here the dark blue points on the left plot are the initial random sample, and the red points are the new batch that we took after running inference with the model and taking the lowest FEP values. So if you look to the right, the FEP values of this new batch kind of shift to the left a little bit, which is a good sign. So the model is, at least right off the bat, finding some candidates that are better than the initial set in some way. We're kind of moving in the right direction, which is a pretty good sign. And we also kind of identified 15 more molecules in the top 100. So already with 1% of the data sampled, we have 16 of the top 100 molecules by FEP, which is pretty fast. So we do this cycle again, we get a new batch. In this batch, things kind of start to cluster together in the top of the search space there for whatever reason. The average FEP of these values is about the same as the last cycle, but maybe we're sneaking a little bit more left, so we're still finding good molecules in general. So we're still making progress. We do it a third time, just kind of the same approach, and we get a little luckier on this one, where the best FEP goes from somewhere around a little bit under -20 to probably closer to a -25 or somewhere in there. The sample's still, concentrated at the top part of the search space, but it's still finding great molecules. So after 3 rounds, 2% of the data has been sampled, and we found 37 of the top 100 molecules. So you can do this kind of as many times as you want. After 5 cycles, So after 300 samples, which is 3% of the data, we were able to recover about 50 or about half of the top 100 molecules. There is also kind of this possibility when you do something like this where there's kind of one cluster of molecules that are good. So the model finds one specific motif of a molecule that's good and just finds 49 molecules that are extremely similar to each other and they all just happen to be good. We wanted to avoid that or see if that was happening. So to try to test that, we kind of clustered the top 100 molecules and it came out to 35 clusters, and then tracked how many of those clusters had a molecule that was retrieved by the active learning process. So 19 out of the 35 or so were recovered after 5 cycles, which was a good sign that the model and the active learning process wasn't just getting stuck in kind of like a local minima or local maxima and hammering the same molecule motif. Cool. So after running one, it's always, an extension just to run more and see what happens. And since we had the ground truth values of FEP, we could kind of identify a few different knobs and things to turn and try out and see what happens and see how it affected kind of the active learning turnout. And so we tried that with 4 different kind of knobs or parameters. The first one being how we initialized the active learning process itself. So we've been talking about the random initialization, which is just take whatever molecules randomly. We tried a clustered initialization, which first clusters the 10,000 molecules and then picks one from each of the clusters. And that just kind of tried to spread out the sample so that we didn't get a lot of molecules from a small area of the search space. And then to try to kind of test the limits of active learning, we did the worst molecules, which are the highest molecules with the highest FEP values, because low FEP values are better. And we also tried the molecules that were furthest away from the top 100 by Tanimoto similarity using molecular fingerprints. Surprisingly, what we found was if you ran it for enough cycles, they all kind of converge. So here at about 10 cycles, they all kind of start to converge to the same performance. But if you do start from the worst or the furthest away set, you kind of hamper the initial performance of the first few cycles. So in some ways, if you get unlucky with your start set, you will kind of find poor performance for a little bit, but the active learning process can recover and rebound from this and eventually start to find good molecules again. We did the same kind of experiments with algorithms. So we tried Gaussian processes, random forests, gradient boosted machines, and simple multi-layer perceptrons. And somewhat surprisingly, we found that this one also had limited impact on the outcome of the active learning process. You can see there's some randomness in each of the runs. So you have these confidence intervals and they all kind of overlap and are bunched together. So at least for simple machine learning algorithms, the choice doesn't really matter that much. Same thing happened for acquisition functions, where we tried a variety of things that put more emphasis on either exploring or exploiting. Greedy being just let's exploit out the knowledge we have and take the best thing that we think, where cluster being let's explore the space as much as possible. And again, somewhat surprisingly, things turned out to be very close together. Even though probability of improvement and expected improvement seem to plateau a little bit, everything kind of ended up in the same range. We did find that samples per cycle had a large impact on the active learning cycle, or the active learning process, as you might imagine. So we varied the number of samples we took at each round from 5 to 100. And intuitively, the more samples you take, the better you did. You just kind of have more shots on goal, is where that comes from. So that was the biggest impact, but it is kind of good to take note that this was a single data set. It could have been an easy data set. The choices we made for these different parameters to try just could have been too similar to each other. There could be other things that do better or worse, or things like that. But the good news is that in all of these cases, we were able to recover a large portion of the top 100 molecules while only seeing, quote unquote, sampling a small portion of the data set. Cool, well, so that's it for me. All the information here and more details are published in the paper if you're interested in learning more. Just want to say thank you to all the co-authors. It's a really fun project for me. And I will pass it over to Pat.
Great, thanks, James. All right, I'm hoping my screen is visible. Somebody give me a thumbs up. Cool,
Yes.
All right. So James told you about how we've applied active learning in a lead optimization context. I want to talk a little bit about how we can also use active learning in a hit discovery context. And I probably don't have to spend a lot of time talking about virtual screening, but just for anybody who hasn't seen this, in virtual screening, we typically start out with a protein structure where we have a binding pocket, and then we're going to computationally screen through large sets of molecules, varying different translational and rotational degrees of freedom, trying different orientations and trying to figure out which molecules will fit into this binding pocket. This is a relatively quick experiment. We can do often up to 1 billion molecules, and it's relatively economical. So, a few years ago, we really wanted to see how much we could push the envelope here and really how many molecules we could process. So this is from a presentation that my colleague Levi Pierce gave way back in 2019, where we set up a workflow on Amazon Web Services. We spun up 100,000 CPUs, and we were able to brute force virtually screen 1 billion molecules. And the cost was relatively low. So compute cost for that was 12,000 molecules, which is great. And we were really proud of ourselves, patted ourselves on the back. But then the world got more complicated. And in 2019, there were about 1 billion molecules available through what's called synthesis on demand. So with synthesis on demand, a vendor has a set of chemistries that they're comfortable performing and confident that they can deliver. You can usually get these molecules for about $100 a piece, and you can get them within 6 to 8 weeks. So it really has revolutionized the way that we can do virtual screening. The thing is, the size of these databases has been growing at a tremendous rate. So instead of having 1 billion molecules commercially available, you now have a database from Enamine that has more than 1 trillion. This Enamine real space that we were working with was 1 billion in 2019. It's now 36 billion. Actually, this slide is out of date. As of a couple of weeks ago, that database is now 40 billion. So, the point I'm trying to make here is there's a lot of molecules available, and this brute force approach that we have just isn't going to fly anymore. So, as I was saying, in 2019, we could do 1 billion molecules, and it would cost us about $12,000, which in a drug discovery program is, not a big deal. But then, that database got to be 10 times bigger, and suddenly it's going to cost me $120,000 to do that computation. And now, as we're getting into trillions, it just isn't economically feasible to brute force virtually screen these huge databases. So how do we approach this? And I think, hopefully, as you saw from the earlier part of the talk from James, you can see that perhaps we could use this active learning method and then start using this combination of exploration and exploitation to approach these very large databases. So I think we're at the point now where we've got to think about how to do things differently. So I want to talk about one technique that we're going to use to approach this very large scale virtual screening problem. And we're going to borrow some ideas from gambling theory. A lot of statistics that we use now is really founded in a lot of earlier work that was done looking at ways to improve your odds when you're gambling. So we're going to use a technique called Thompson sampling that is an implementation of what's called the multi-arm bandit problem. So in the multi-arm bandit problem, I have a set of slot machines and I can pull levers on those slot machines. But how can I come up with the most optimal order of pulling those slot machines to maximize my payoff? So one way to look at that is to use Thompson sampling. So I'm just going to give you a simple example. Let's say that we have 3 slot machines. I haven't pulled any of the slot machines, so I don't really have a clear idea of which one has a higher probability of paying off. So what I'm going to do anyway, and then if I look at the probability distributions associated with those slot machines, I haven't pulled them. So for all 3 slot machines, my distribution is uniform. So what I'm going to do now is just generate a random number from each of those 3 distributions, and I'm going to pull the lever on the slot machine for the machine that has the highest random number. You see that'll be the one in the middle. So I'm going to pull the lever on that slot machine. So that's going to change the distribution associated with that particular slot machine. I had a winner. So since I won with that pull, that's going to adjust the probability distribution associated with that machine. So then if I generate another random number, you'll see that this time the distribution in the middle is different. So I'm going to get the largest random number out of the middle machine. I'm going to pull that slot machine again. This time I lost. So now I have a normal distribution associated there. So again, I'll do another random draw. I'll pick a random number from each of those distributions. Based on those pulls, I again get the middle slot machine. So I'm going to adjust the distribution associated with that middle slot machine. You'll see that now the distribution shifts a little bit more to the right. So I'm going to be more likely to pull the highest random number from the distribution in the middle, but I can just continue to repeat this process of generating a random number from the distribution, selecting the one with the highest random number, pulling that slot machine. And I'm just going to keep doing this and keep making pulls. And you'll see that each time I pull that slot machine that's associated with the highest random number, I am going to adjust my probability distribution. So as I keep going through this and making these pulls, you'll see that I'm getting more payoffs and I'm adjusting the distributions that are associated with each of these machines. So now those of you who are astute observers are saying, this is great. What the hell does this have to do with virtual screening? So let me see if I can now take the setup that I've done and explain Thompson sampling and show you how we can apply this in a virtual screening context. So first of all, let's think about these molecules that we're screening. So these molecules are created typically out of simple 2 or 3 component chemical reactions. So if we look at this set of 9 molecules here, then you can see that these 9 molecules are actually made from reactions of 3 building blocks at position 1 and 3 building blocks at position 2. So the 3 times 3 gives you 9 products. So if we start thinking about this in terms of these large libraries, if I've got 1 million molecules, that's 1 million molecules that I've actually made out of 1,000 building blocks. So rather than searching this space of 1 million products, can I do this and reframe the search in terms of 2,000 building blocks? So if I have a space of 1 billion products that are made out of 3 components, instead of searching that space of 1 billion products, can I turn this into a space of 3,000 reagents? So searching the building blocks rather than the product. So let's just take a look at how that would work. So I could start out again, like we did in the gambling case, where let's say I start out and I know nothing. So what I'm going to do is I'm going to pick one of my reagents R1, pick one of my reagents R2 at random. Then I'm going to put those together to form a reaction product. So now I've got a larger molecule that's the product of that reaction. Given that reaction product, I can then convert that molecule into a set of 3-dimensional structures representing the flexible conformations of that molecule. I can then take each of those conformations, put it into a protein binding site. Then what I can do is I can score that, and I can get a score associated with that molecule. Much like what James showed you, a more negative score is going to represent a tighter binding molecule. But now that I've got those scores associated with reagents, I can work backwards from that structure back to 2 reagents. And I can associate and propagate the scores back to those reagents. So now coming back to Thompson sampling, what I'm going to do is I'm going to then, as I sample, I'm going to associate that distribution of scores with each of these building blocks. And then I'm going to let those score distributions determine how I sample the building blocks and which product molecules I'm sampling. So, how well does this work? So as I mentioned earlier, we had done these brute force experiments where we've already done virtual screening on data sets of up to 1 billion molecules. But for this particular example, we'll take a look at this set of 400 million molecules. So we brute force docked 400 million molecules. We used Thompson sampling to sample 0.5% of the database. And now what we're going to do is we're going to compare the score distributions from 10 different Thompson sampling runs versus the ground truth, which is from the brute force docking. And what you can see over on the left of this plot in pink, that's the ground truth. So that's from doing the brute force docking. Then what we have in blue is 10 different Thompson sampling runs. And you can see that from these 10 different runs, in 8 out of 10, we found the best molecules. And then if we combine those 10 Thompson sampling runs, you can see that the distribution that we get from the Thompson sampling is actually very close to the distribution that we get from the brute force virtual screening by only screening 0.5% of the final database. So let me show you just one example of how we put this into practice. So this is a really nice collaborative effort that we did with Jamie Fraser, who's a crystallographer at UCSF, Brian Schoikert, who's a computational chemist at UCSF, and a lot of this work, the crystallography was done by Frank van Delft and his group at the Diamond Light Source in the UK. So what we're doing here is a fragment screening approach. So what we do is we take a library of a couple thousand very small molecules. So these are molecules with probably on average 10 or 12 heavy atoms. So you're taking these very small molecules and one at a time, you're crystallizing those with a protein and looking at how that molecule binds to the protein. So what we're looking at over here on the left is an overlay of 210 different structures. So from those 210 different structures, we can see how all of these small molecules, these fragments, bind to the protein. Now, one thing to note is the binding of these is relatively weak. So most of these bind with millimolar potency. So really, to get to the point where you have a drug, you would have to improve the potency of these by about 1,000,000-fold. So how do we do that? And typically, there's two ways that you do that. One is to take that fragment and grow it. So you try to attach other chemical moieties to that, expand and make that fragment larger. And then by making it larger, you're making additional interactions with the protein, and you're going to make it more potent. You're going to make it a tighter binder. The other one is you could take these pairs of fragments, and then you could try to come up with chemistries that will link them together. And then in some cases, when you link these, you'll again get this multiplicative increase in potency. The thing is that this is really tricky, because unless you get that geometry perfect, you'll typically disrupt one of the two binding partners. So we decided to take an alternate approach. So you remember, we've got 210 of these fragments. So what we did was designed an approach, and this is an approach in our group that was pioneered by Molly Schmidt, where we take these pairs of fragments, then we do a search through these large databases to try to find something we can buy that overlaps with both of those. So I want to find one molecule that overlaps with those two. And then rather than doing a lot of chemistry, I'm just finding these molecules that I could buy for $100 a piece. Now, we don't really want to consider all pairs of ligands, because some of these are just too far apart. So we picked pairs of ligands where their centers were within 20 angstroms of one another. You don't want things like cases 1 and 2 here, because those are sitting on top of one another. So the combinations are not going to be interesting. So we also filtered based on just the volume overlap, and we didn't want things that overlap. But this was still a lot of queries. So when we looked through all of these and we started filtering based on things that were, close enough, but not too far away, but didn't overlap, that gave us almost 5,000 queries. So we wanted to run 5,000 queries against 23 billion molecules. It just wouldn't work to do that. It would have taken years and years, and the compute would have been incredibly expensive. So rather than doing that, we used Thompson sampling to search through this. Using a Thompson sampling procedure, like I showed you earlier, we went through and selected 39 molecules to have synthesized. Out of those 39 molecules that we had synthesized, we were able, or our collaborators at UCSF were able to get structures of 12 of those molecules. And we found that we got about 1,000-fold increase in potency. So we went from molecules that were double-digit millimolar in terms of potency down to things that were double-digit micromolar. And here are some of those molecules and some of the crystal structures. But the really cool part about this was we got the right answer for the right reason. So if we start out with the original fragments that we were working with, then we used those fragments, we directed a search, we found molecules like the one in the middle, where you can see how it overlaps with those two fragments. And then when we got the crystal structure, so this is the one in the middle is a prediction of how the molecule would bind. On the far right, we have the actual experimental data. When you put those on top of one another, you can see that our prediction for the binding pose was correct. We did another round of that, and now we've gotten to, by purchasing fewer than 100 compounds, we've now gone from compounds that were 5 millimolar or 10 millimolar down to molecules that are 5 micromolar. So we've got nice, efficient molecules. We've been able to, with a relatively limited computational and experimental expense, get essentially a millionfold increase in potency. So, hopefully I've shown you through this part another way that we can use active learning that's complementary to what James showed you. So, as James mentioned, we're using machine learning across the drug discovery process, hopefully just showing you a couple of examples of how active learning can be used. As I see it, this is still just one piece of the puzzle, and you really need to have this combination of experimentation and computation to drive your drug discovery project forward. Finally, want to thank everybody who worked on this project, the folks at Relay Therapeutics who were part of this collaboration, all the work that was done at Google, and then our experimental collaborators at UCSF. Thank you for your attention, and we'd be happy to take any questions.
Perfect. Thank you so much, Pat, for the great talk. If you have any question, just raise your hand and jump right in.
Yeah, I'm just kind of looking in the question. So, can Thompson sampling be applied to an ultra-large, non-enumerated chemical space? And that's one of the beautiful things about this, is the way that we run Thompson sampling, you don't have to fully enumerate the space. So, if you think about it, if you got 50 billion molecules, just think about how much, that's probably a petabyte of disk space it would take you just to hold the conformers. So the great thing about Thompson sampling is we do all of this on the fly. I see a few people with their hands raised. So Prudenzio, do you want to be the
sure.
the moderator, or should I? Or
sure. Okay, we can start with Aish.
Hi, great talk, Pat and John. So I was just wondering what kind of the protein binding sites, is this technique sensitive to what kind of sites is it, or would it work well on any kind of binding sites?
I think it would work just about, there's been some work from Brian Schoikit's group at UCSF where they've done it on GPCRs. We've done similar things on a variety of targets. So yeah, I think anything where you feel that you've got some confidence in the protein conformation, I think you're in a pretty good place.
To grow, and one related question, to increase the binding affinity from from milli to micro, what technique did you really use? You mentioned there were a bunch of options. So the one you used was growing them, like adding kind of.
No, the one that we used is roughly equivalent to linking, but rather than trying to say I've got fragment A, I've got fragment B, and I want to find a linker to put between them, what we do is look through these large databases and say, can I find something that overlaps with these two fragments? So it's really more of an overlap technique.
Okay, thank you.
And there's another question. Why did the molecule data sets start to explode over the last couple of years? And I think it's because people figured out you can make money doing this.
Yeah, I do have one question, but have you tried to use this Thompson sampling approach to go from micromolar to nanomolar binders? Like when the bar is higher in terms of binding? I mean,
You know, I think when we're trying to go from micromolar to nanomolar, what we're typically doing is we're moving more into the realm that James was talking about, where we're using FEP. But yes, we have also done work to couple Thompson sampling with FEP.
okay, and does it work as well as just using a plain native learning loop?
Yeah, it does.
Okay, perfect. I do have one question for James about his part. When you were sampling many molecule per cycle, I was wondering how do you balance, like, because like you're sampling different molecule, I'm sure you are not just taking the ag mass of the acquisition function, right? So how are we balancing the sampling, like diversity and like exploration and exploitation within that batch?
Yeah, I mean, a couple of different ways to do it. Sometimes we did just take the argmax of the batch, and that seemed to work pretty well. But you can kind of scale that from 100% greedy to 100% explorative, and kind of shift that shifter. And we found, at least for our data set, it doesn't have too much of an impact, but that could be different for different data sets or approaches or things.
Yeah, that was one of the motivations behind this work. Was other people had applied active learning, but they had used these really elaborate acquisition functions. So, the 2 rounds of exploration followed by 2 exploration, followed by, a clustering step. And there was really never any rationale behind that. So one of the things we wanted to do was generate this large data set and then investigate a variety of different schedules of acquisition functions.